welcome to this class on Neuroscience of Human Movement. So, this is part 3 of our discussion on skeletal muscles. In this class, we will be talking about how in a muscle force and length are related and force and velocity are related, or in other words, here when we say length, we refer to the length of the muscle, and when we say velocity, we refer to the rate of change of muscle length. So, in general, velocity means there is actually an observed displacement, right. So, I am making a movement like that, I am making a reaching movement, for example. Here when I do that, there is a velocity of movement, but that is not the velocity that we will be discussing in today's class. In today's class, we will be discussing the rate at which the muscle length changes. That is what is called as velocity of shortening for a muscle, and how this velocity of shortening of a muscle interacts with force is the other point that we will discuss. So, we will be discussing force length relationships and force velocity relationship. Essentially, this is the curve of length. So, say this is in microns, and this is force as a function of the maximum force that can be produced, which is why it is a percentage. So, there is no unit. This is force that is a function of the maximum force that can be produced. How are the two related? This is how they seem to be related, like that, right. Now, it makes sense for us to think about as the length increases, the force increases. That happens, for example, as the length increases, the force seems to decrease. We will discuss all the consequences of this in today's class, right. So, what happens at this stage of the, or in this regime of the force length curve, right. So, we are talking about that regime. What happens in that regime? That is what happens. So, to zoom in, what you see is that the blue colored filaments here are of course myosin, the red colored ones are actin. In the classic model of the sliding filament theory or the cross bridge theory, we saw that actin is hanging in from one end and hanging in from the other end, they do not actually touch. But when the length becomes too close to each other, then what happens? Actin starts overlapping with each other. So, approximately here this is the end of one actin filament and the other actin filament, but they both start overlapping, producing an overlap here. This causes a situation in which the myosin cannot bind with the maximum number of actin sites, right. So, this reduces the force that can be produced. Now, if the sarcomere length increases, what you would expect the actin filaments to move farther away from each other, right, like that. So, here is the tip of the actin filaments, right. Now, around here the force increases as the length increases. So, the length has increased from that point to that point. So, that is the change in length, whereas the change in force is that much. Okay. All right. So, now again now there is a greater possibility that myosin gets access to actin binding sites. Now, I can further do this, increase the length, then what happens at around that point, I am going to have a situation where there is maximum actin myosin overlap. Let us remember that it is the interaction of myosin with actin that causes force production in the sarcomere and in the muscle, right. So, for us to have a large amount of force, for us to have maximum force, you have to have an optimum length at which myosin is able to interact with the largest number of actin filaments. But not just that, it is also that at shorter levels the amount of effort that myosin will have to put to recock, attach, detach is higher, because there is very little space available. Usually it also takes more time. So, some other phenomena other than just actin overlap are involved, but then at the maximum force level what happens is that there is maximum overlap of actin and myosin filaments. This causes maximum interaction of actin and myosin, thus also producing a maximal force. So, that is the 100% force. As I said earlier, this is the normalized force. How is this normalized? This is the force produced divided by the maximum possible force produced, right. That reaches 100%, or the maximum force is actually being produced. If that is the case, you would expect, well, maybe I can keep increasing the length, then I can have, instead of this I can keep increasing the length, I can keep increasing the force too. Well, obviously force cannot go above 100%. What happens at this stage or at that regime is this, right. See here, some myosin molecules do not have interaction sites. So, these are not able to interact with actin. Why? Because the length has increased. Further, the length has increased more than the optimum length. Because of this reason, whatever the whatever actin molecules that can actually interact with myosin, these only are responsible for force, right. So, this reduces the amount of force that can be produced. Then what happens if I further increase the length, right. So, the question is what happens at that regime. At that regime what happens is that there is very little, if any, overlap between myosin and actin. Actin again is in red and myosin is in blue. You see that there is very little overlap, that means that the force that will be produced will be very small. So, there is a particular point at which actin and myosin are overlapping at an optimum level. This is the point at which the sarcomere is going to produce its maximum force, right, which is 100% of its 100% of the absolute, right. So, in this case, right. So, this is the reason why you have a non-monotonic relationship between force and length in a sarcomere. As the length increases, the force increases until some point, like it happens in a spring, right. Like you have stiffness, the slope of this relationship, right, indicates some form of stiffness, right. So, I could have, except let us remember that for a spring F is equal to kx, right, where F is the force and x is the displacement, k is the, here this is a linear relationship. However, in the muscle this is not going to be linear, but it is not just that, muscle is not just a non-linear spring, it is much more than that. In general though, many times in biomechanics people use the slope of the force length as some form of stiffness, sometimes it is called as apparent stiffness. When the force produced by the sarcomere, when it is greater than the external load on the muscle, what will happen? Will reduce, is it not? Why? Because it is sarcomere force that is greater. Suppose the force produced by the sarcomere is less than the external load, and this is not exactly uncommon. Sometimes the external load is higher than the force that can be produced by the sarcomere. It need not just be the external load. For the purpose of this discussion, when we are looking at one sarcomere, the force produced by another sarcomere can be acting as a load for this sarcomere, for the first sarcomere. So, some load, that is if the force produced by the sarcomere is less than the load on that sarcomere, then what happens, the sarcomere length it increases, right. So, you can produce force both when the sarcomere is contracting or when the sarcomere is undergoing contraction and it produces a force. You can also produce a force when the sarcomere is lengthening or extending, right, and it produces a force. These two forms of contractions are also called as concentric contractions or shortening contraction in the first case, and in the second case when the sarcomere is lengthening and it is producing a force, it is called as lengthening contractions, or sometimes also called as eccentric contractions. We will discuss that in a future class in some detail. So, let us remember what limits the velocity, right. The maximum rate of change of sarcomere length is limited by this biochemical process, right, by the interaction of myosin with actin, the speed at which cross bridges can form. So, there is attachment of myosin to the actin, and there is a pull, there is power stroke, and then there is detachment, and then there is recocking. There are a bunch of steps in the cross bridge cycle or in the sliding filament theory that will have to be repeated. The speed at which these steps can happen, right, the rate at which the cross bridges can be formed limits to what extent the muscle can contract, to what rate the muscle can contract, right. So, the velocity of contraction is limited by the rate of cross bridge formation. Okay. Now, let us take a look at that situation, right. And also in summary what happens at various lengths at a very at 100% force or at the optimum length. This is also usually the resting length of the muscle, but not always. Often this length is the resting length of the muscle, but when I say often, it is not always, there are exceptions. At the maximum force, you are going to have maximal overlap between actin and myosin. At a length that is smaller, so when the length increases, what happens is that some myosin molecules close to the middle of the sarcomere are not able to find binding sites on actin. So, they are not able to form cross bridges and produce force. So, the force reduces at this point from here to here, right. And then as the length continues to increase, there is very little or no overlap between actin and myosin, thus resulting in a steep drop of force. So, deviations from the optimal length cause deviations in force, and this deviation in force is relatively small at small deviations in length, but as the length deviation increases, the force deviation increases much more than one would expect. So, this is minimal for small deviations, but the tension drops off much rapidly. So, at a much higher slope much rapidly as the length deviates further from the optimal length. The active force is shown here in brown, the passive force is shown in blue, the total force is in green. Until some point, until about that point, right, until that point, the brown and green overlap here. In this region, brown and green overlap, or in other words, the total force is basically the active force. So, at that point there is a development of a passive force. Why is this passive force getting developed? Because it turns out that muscles are composed of contractile proteins, like for example, a rubber band type of situation, right. If you have a rubber band, when the rubber band is slack and you are trying to increase the length, as I keep increasing, there is going to be a particular point after which if I increase the rubber band's length, it is going to start resisting changes in length, right. The length at which it starts to resist changes in length or produce a passive resistance, that is called as the resting length of this rubber band. Likewise, you are having contractile proteins in muscle that start resisting the force, and that is shown here as approximately that value. At around that value, the muscle also starts resisting passively. So, there is passive response. There is also active response, but then the active response seems to drop off after some time, then the passive response starts increasing. Combined together, basically there is a relatively strong resistance beyond a particular length, right. Beyond an acceptable length, there is going to be a great resistance, which is basically a combination of both active and passive resistance. What do we mean by active resistance? Active resistance is the resistance that is produced by cross bridge formation, by neural control. So, basically neural control to neuromuscular junction to excitation contraction coupling to cross bridge formation. It is this that causes the active force. Passive force on the other hand is caused purely by the passive properties or the contractile proteins that form the muscle. Now, so let us remember, at any given point in time, if I only can measure the total force, it may not be possible for me to estimate. So, at any given point in time, if I am measuring the total force, it is not possible for me to estimate how much of this is coming from active force and how much of this is coming from passive force. Many times this is of importance in biomechanics, this is to be estimated in some cases. So, this varies as function of several factors. Okay. Now, the question is what happens to velocity? Well, what happens, we can discuss this before we discuss the plots, we can discuss a situation. Right. I am having a relatively light object in my hand. So, what you would expect is that it is going to be possible for me to lift this with a very high velocity, is it not? I can do this. Suppose I had a very big mass. One question is whether I will be able to lift it. Suppose I am able to lift it, it is immediately obvious that I will not be able to lift it at that very high velocity, right. Time rate of change of sarcomere length is what is called as velocity of shortening of a sarcomere. So, that is also inversely related to the force, like it happens here, that is also inversely related. So, what happens is the velocity is practically 0 at a particular value of force. This is also called as the maximum isometric force that can be produced, right. Let us remember what is the isometric force. Isometric force is the force at which the length remains the constant, is it not. So, beyond this level, so this is the shortening regime, and here what happens, what is this? So, can the velocity be negative? The answer is yes, because it can also be lengthened by external forces or stretched, right. When that happens, a different maximum force is produced. So, this is, I am going to call that force as the F max isometric, and this is the F max absolute. This is the absolute maximum force that can be produced, that happens as the lengthening increases. But then as I want to change the velocity, as I want to increase the velocity, the force that can be produced drops down quite quickly, right. So, why does the force reduce? As I said, the speed at which cross bridges can be formed reduces basically. There is reduction in cross bridge displacement, and there is also a reduction in the ability of myosin to find active actin binding sites, right. An important point to note is that the preceding activity affects how the muscle behaves, right. In other words, if before this experiment the person has done some exercise, right, then the muscle will behave in some manner. If the person has not done any exercise or is at rest, the muscle will behave in a different manner, right. In general, if there is an activation before the experiment, there is going to be an increased ability of the muscle to show stiffness, or there is an enhanced stiffness, but this does not last for a long time after the exercise. So, this increase that is temporary, transient, post exercise is called as short range stiffness, right. So, also what happens with power? What is the relationship between power and force? Basically, power is the product of force and velocity, is it not? So, that increases until some velocity and then it decreases. Right. So, essentially these two relationships, what are these two relationships? Force length relationship and force velocity relationship dictate to a large extent how forces are produced within a muscle, within a condition, depending on the situation. They have very important implications and consequences in biomechanics. Examples are limiting the running speed. Say at Olympic level, there are limits to how fast the person can run. So, limits of running speed or limits of strength, how long you can jump, how much you can jump, or how far can you jump, and to what extent you can jump, and with what velocity you can run. All these things are to a great extent limited by force velocity and force length relationships. These can be modified, of course, these can be modified. The hypothesis is that they can be improved, but only till the physical and physiological limits, is it not? The question is in Olympic level athletes to how much you can improve this, and are there genetic influences? That is the other question. So, basically it is possible that a given person can only improve so much. It is a function of age, genetics, practice level or expertise level, and of course, it is probably a function of the psychological state also. Many of these things have been studied. There is data suggesting that specific ages. So, obviously one would expect that a younger person would run faster than an older person, etc. Especially as this age gap increases, the difference in velocities are also going to probably increase. There are exceptions to every rule, because there are old people who are relatively fit when compared with young people who are relatively unfit. So, there is exception to every rule I am speaking about, but these are some of the factors that affect this. So, with this we come to the end of this discussion. We will continue our discussion in a future class. Thank you very much for your attention.